So the last one is really about the gene regulation. So before moving to the gene regulation, let's give you like why we want to study the gene regulation. So in the first three sections, we will already learn lots of things, right? So then the function of this gene that can be regulated by the promoter enhancer loops. And also like there are some like chromatin structure, they can change the like activity of the gene. However, all those changes are really happened within the cell. The pathways that can like read the changes outside of the cell usually is through the transcription factors. So that's why in many cases people are also very interested about like how those transcription factors will determine the expression pattern, and that's why we want to study the so-called gene regulatory network. So why we call it a gene regulatory network? It's like this. So on the whole genome, there are lots of genes, right? But some genes, like when it expressed, it becomes a protein, and this protein can bind to the DNA at another location, and this binding can somehow like determine the expression of another gene, right? So that's why you say like for gene A, it can regulate B, or in this case, like gene Y can regulate gene X. Yeah. So this so-called gene regulation. So of course you can imagine like because it leads to like a transcription, translation, and become a protein and goes back. So there's a delay during this process, during this like whole process. But as I mentioned, this actually is the main like determinant of the cell system. Like usually when cell responds to the environment, it changes on the transcription factor network. The whole network is very complex because there are so many genes. So that's why I first give you a simple example, the network in the E. coli. So E. coli is a very small bacteria. So there are only like several thousand genes. And among those several thousand genes, there are 208 transcription factors, right? So, and then those transcription factors can somehow like trigger the expression of some other genes. So for this very simple network, what does it look like? So it looks like this. So you can see like, of course, because this like transcription network is so important. So that's why wherever like people draw this network, they also try to annotate the network. Like you can see like a different network somehow relating to the function of these cells. But whatsoever, like this is E. coli, right? It's a very like a simple species. Then how about human? So the human regulatory network is something like this. So it's like a very complex network. Of course, again, like people try to annotate the whole network by give them like by using say that it's corresponding to like different pathways, different like activities or different responses. But you can see that it's very complex, and I think that's the reason like we have a very like a vivid life. Right? Because if we only like depends everything on the 3 billion characters, then we cannot have so many like possibilities. I think really is the complexity in this regulatory network can like build such like different like living stuff. So I don't want to like show you like too many things about the network because it's just so complex. But probably like I can show you some examples like why we have some network structures, right? So let's make the problem easier. So for example, if we have only 3 genes, then we have different ways to connect those 3 genes, right? So for example, we can using X to control Y. Like if there are an expression of X, then X will become a protein and then bound to the location Y, and then Y will transcribe, and Y then bound to location Z, and Z will transcribe, right? So they have those like relationships. And those relationships you can see sometimes it's like to amplify the expression or to depress the expression. So that's why we have different arrows. But whatever, this kind of structure is so-called called feed-forward loop. So I will explain more later. So the second kind of, the other kind of structure is so-called a single input module. It's like we have X is a transcription factor, and then it can control the expression of many other genes. like as shown here, it's like Z1 to Z and many other genes. The last case is really like the most complex case, is like many to many interactions. Let's start with the easier case, like the feed-forward loop. So imagine that we have this like AND gate feed-forward loop. So the AND gate meaning that if we have X, then after some delay, we will see Y, right? And if X and Y both present, then it will start the transcription of Z, right? So this is the structure of this AND gate feed-forward loop. Then why we want this feed-forward loop, right? At least there are two like important attributes of this feed-forward loop. The first thing is actually this can be work as a filter of the signature or the signal. So for example, if the X is like receive, they receive a signal from outside, like just sense like how many resources outside in the environment. So if there is a sudden change in the X, you can see because of the delay, like if we have X, then there is a delay in producing Y, right? So then for the Z, because it requires the presence of both X and Y, and due to the delay, then in the whole like time frame, we can only have the X. Then if the signal of X disappear, we have Y, right? Then in the whole process, we cannot trigger Z, right? Because in the whole process, we only have X or only have Y, right? So it's like a sudden change in the environment. So this kind of structure will just say that don't care those like sudden changes. Probably is not important, right? So it can totally ignore such noises in the environment. Well, if really there are like important changes in X, like the second case, there is a constant high expression of X, meaning that something important happening. Then you can see like this X will producing Y, and then X and Y both present, then we will trigger Z. Probably is the correct response that the cell will behave. And a lot of good thing about the feed-forward loop, feed-forward loop is that because it requires both an X and a Y to present to trigger Z. So if X disappeared, then the whole system will shut down immediately. So as a comparison, if it's like only X control Y, Y control Z, then always there is a delay, right? If X disappeared, then they wait a while for Y to disappear. Then for Z, it's like even later. So that's why there is a delay in the whole system. Well, for the feed-forward loop, the cell can shut down the whole system, just as long as they cannot see X, right? So that's why it's a lot of good property that it can rapidly shut down the system. So you can see, even though this very simple like a network structure, they have good properties, like useful properties for like survival. A lot of simple structure is this like a one input and a multiple output system, right? So why sometimes we want this? So you can imagine like genome, there are so many genes, right? How we can, but in many like experiment, like chemical experiment, there is a order to produce something. Like sometimes we have to like express X and then Y. So in this case, like have to express Z1 and then Z2 and Z3. There is a order, right? Then how you can like control this order using the DNA? How you can control this? So the one way to control this is to using the X to control Z1, Z2, and Z3, and then because you know the transcription factor, they have a binding motif, right? So the binding motif meaning that on the DNA, some DNA patterns they have a stronger binding, some they have a weaker binding. So that's why for the cell, they can really modify the DNA sequence to make it a stronger or lower binding during the evolution. So if it's a very strong binding, then just a small amount of X will trigger the expression of a gene, right? So that's why you can see GY properly have a motif that have a strongest binding to X. Then whenever there is X, very low level X, it will trigger Z1. For the Z2, then it have a slightly lower binding affinity. So that's why you need to slightly more X in order to trigger the expression of Z2, right? The same thing for Z3. So it means that there is a way you can just change the DNA to change the order, how you can express all those 3 genes or even more genes using this lateral structure. Yeah. So that's why even though it's a very simple structure, sometimes it's very important. And that's why we can using this one-dimensional thing to encode such a complex logic in the cell. The last case is just basically a summary of what's the potential of different combination of complex structures. So for the interest of time, I just skip it. So it's not very important. So as a summary, so I think today I've shown you what's the 3D genome structure, right? So at the basic, we have the enhancer loop interactions. And then on the global picture, we can view the heat map at a different resolution. On the very low resolution, they are open and closed region. A little higher resolution, we can see different domains, right? Different triangles. And then if we go further, then we see enhancer promoter loops. And many of the loops are controlled by the CTCL binding domain. If we disrupt the domain, then somehow we disrupt the loop. And then the last part of the lecture, I tell you a little bit about the gene regulator network. Is one protein can control the expression of another protein. And for this controlling, we have a different motif, lateral motif, and sometimes this lateral motif can be very useful to perform different functions, like preserve the temporal information, like the order information for the cell system. Yeah, so I think that's all for today's lecture. So thank you. Yeah.